Well, good afternoon. This is going to be a short video. Probably should only take about 20 minutes for me to go over phylum Cnidaria or phylum Coelenterate and Porifera. Phylum Cnidaria and Coelenterate are used simultaneously or one in place of the other because basically it's the same thing. And phylum Cnidaria basically are consisting of hydra, jellyfish. We look at coral and sea anemones and the Portuguese man-of-war, sea plumes, sea fans, stuff like that. So that is all encompassed in this particular section that we're going to talk about. There is a basic, the body structure of the phylum Cnidaria basically is polymorphism, which means that they have more than one body form. They're either a polyp or they're a medusa, and we're going to discuss the difference between the two in just a second. But just so that you understand that a polyp is adhered to the ocean floor or to rock or something, and it has its tentacles facing up, and the only hole that is on the polyp is on the top, while a medusa is what you would normally think of as a jellyfish. Bell-shaped, tentacles are facing down, and the only hole it has is on the underside of the medusa, or that bell shape. The phylum Coelenterate and Cnidaria basically have a gastrovascular cavity. So what that gastrovascular cavity is a hollow intestine, basically. So as food goes in, enzymes are secreted, it is broken down, and waste goes out the same hole it came in. So the mouth and the anus, when you see the diagram of a polyp or a medusa, and you see that it is the same hole. So a polyp we think of as a cylindrical shape, and it is basically a tube with tentacles around the mouth, and it's sessile. Remember we talked about sessile being that food has to come to the animal and the animal is not going to the food. However, it is using its tentacles to grab food toward it, but it's just not moving as far as what you would think of an animal as moving. So when we think of a polyp, you could think of like a sea anemone. The structure of a sea anemone is a polyp. Coral is a polyp. A hydra is a polyp. Your book talks about hydra. Hydra is found basically in fresh water, not in ocean water. And let's look at medusa. So the medusa has the umbrella shape, and the medusa has tentacles that are facing down, and the medusa is motile, it is free swimming. These tentacles, both on the polyp and the medusa, have what we call cnidocytes, and cnidocytes are responsible for the stinging cells that these animals have. So Cnidaria just means animals with stinging cells. Those stinging cells are called cnidocytes, and what actually does the stinging in that cell are the nematocysts. Okay, this is all in section 21.3. I'm just trying to give you a quick overview of it. And again, you can pause this video at any time and just take notes. But the spelling is there at the end of your chapter under the defining of the words. You can just look at the spelling there and go ahead with the definitions that I'm giving you as we move along. So basically, when we're looking at the tentacles of either the polyp or the medusa, we can say that they have those nematocysts, right, which are the stinging cells. And basically within the cnidocyte, within this stinging cell, excuse me, there's this coiled thread, and this coiled thread, if you can think of it almost like spring-loaded, okay, this coiled thread is is discharged when the organism senses either food is around it or it feels threatened. Okay, this coil thread discharges like a harpoon. Okay, and there's like a barbs on it, and that is what will grab hold of the prey and hold it, and it releases neurotoxins. So these neurotoxins will paralyze the prey. Cnidarians don't really have they have specialized cells, but they don't have really a defined organ system like you would think of as those in the animal kingdom haven't. So when we look at cnidarians or coelenterates, we can say that they have radial symmetry. And remember, we've already gone over radial symmetry, bilateral symmetry, and asymmetry, so you should be able to have a clear understanding of what radial symmetry is at this point. The habitats of the polyp and the medusa are aquatic, mostly marine, but the freshwater one we will discuss is the hydra. And they are carnivorous, so they are predators. They are going to eat other animals, and again, they get that by using their tentacles and the nematocysts that will then shoot out like the harpoon and grab its prey and paralyze it. And the tentacles are used to grab that prey and then to bring it to its mouth. The gastrovascular cavity that we had already talked about does make enzymes, and it allows for extracellular digestion. And yeah, just an overview there. Let's see here. Let me just see if I can't get. Nope. Not letting me. I wanted to see if I couldn't get this up on the screen big enough for you to be able to see. No. Hold on a second. I'm trying to show you a picture of a jellyfish here just to be able to go over some of its structures with you. I'm going to have to big and view. Oh, I think I did that wrong. I did. Alright, I was trying to get a picture of the jellyfish. Maybe I can do this. Alright, so when you see the jellyfish there, you can see the tentacles hanging down here. Alright, you can see, you really can't see a mouth opening, but you would have to know that it's basically at the center in between that. And you can see how just long these tentacles really are. Okay. And that was a medusa, obviously, because it was bell-shaped. And what happened there? Alright, so... Alright. They respond. Jellyfish have what is called a nerve net. Okay? They don't really have a central nervous system like we think of a central nervous system. They have a nerve net, and that nerve net gives them sensory cells that allow them to respond to chemicals in their environment, touch, light. They do have an eye spot, which is ocelli, and statocysts actually allow them to help with balance. How do they move? They actually move. We said they were free. The medusa are free swimming, and then the polyps are sessile. But the exception to that is the hydra, and the hydra are actually they tumble on their tentacles, and again they're in fresh water. And sea anemones tend to glide on a petal disc. So there's just some differences there. So reproduction. with the cnidarians happen either asexually or sexually. If it's happening asexually, it's basically there are budding actually forms on the polyp or the medusae, but the budding actually forms on the polyp, and it will grow and it will produce a new polyp. And let's just see here. If you would turn to this section of your textbook here, you'll see the reproduction of the medusae and the polyp. In sexual reproduction of the medusae, it releases sperm and egg, because there's male and female jellyfish. And what do you call it? They release it. It basically attaches to the mouth because, again, it's the same hole. And they will then, the egg, once it is fertilized and is a zygote, it will, or larvae, it will release from the mouth. It will swim away, and when it's done swimming, it attaches to the ocean floor or a rock, and it will grow. As it grows, it's a polyp form, and as it's a polyp form, it will mature, it will bud, that bud will break off and form a new polyp, or if it has the opportunity to grow and mature, it forms medusae, and the medusae then will actually break off of the polyp, and this is very microscopic, you're not seeing this, but it will break off, and then it will swim away, and as it grows, it will become a full-fledged jellyfish. So when we look at medusae, we can say, oh, I'm sorry, cnidarians, we can say that they have an ecological role in our environment. They prey on fish and microorganisms. Their neurotoxins are used in medical research. Coral is actually used in jewelry, in buildings, and in reefs for surfing. And coral reefs are a habitat for many aquatic life. They have great biodiversity, and they protect the coastlines. And there is symbiosis with other organisms. So again, there's a relationship that either one or both are going to benefit. Let's see here. Let me just see what my other notes have. So, yeah, when we look at that symbiotic relationship, sea urchins can live in anemones. I'm sorry. The clownfish also live with the sea anemone. Let me see. I think that is it basically for the cnidarian there. So when we look at, I was going to say, when we look at Hydra, the same thing. They are going to, we said, move around on, like, tumbling on their tentacles throughout the water or whatever. Hydra are very small. We basically give credit to Leeuwenhoek for being able to find these organisms in his pond water studies. They will use their tentacles not only to move around, but they will use their tentacles as well to paralyze their prey and to be able to eat it. And again, there is a picture online that I did see that showed like the tentacles squeezing a water flea, and then it is actually able to ingest that water flea. And if you look at that, it looks enormous, because the water flea looks so much bigger than the whole of the Hydra, and yet it's able to consume that. So that's pretty much cnidarians there. If you have any questions with that section, you can go ahead and give me a call or email me. So let's look at the structure of porifera and sponges. Let me just pull up my notes here. That's not it. Hold on one second. I'm sorry, guys. I think it's here. Yeah. So, sponges, they also look like plants, and so at one point it was understandable why biologists would have thought that they were plants, but today we know that they are a sponge. It's a simple organism. It is multicellular, and we're studying kingdom Animalia. We're looking at invertebrates, and specifically we're looking at phylum Porifera here. Sponges have specialized cells, but again, no tissues or organs. Most but not all sponges have asymmetrical symmetry, so you can see that there's a difference now between the cnidarians and the sponges here, or the coelenterates and the sponges. Sponges, like all animals, are eukaryotic, meaning that they have a nucleus, they have inside their cells. And the peripheral word in Latin, it just means pore, P-O-R-E, bearer, and it refers to the organism as having many pores. Cells around the pores are called porocytes. So again, cytes meaning cells, C-Y-T-E-S. All right, cell pores. And because of these pores, a sponge can soak up and release water. So at one time, these sponges were used to actually clean, but today we have synthetic or man-made sponges that are used, but people definitely did use these animals to clean with. All adult sponges are sessile, meaning that they again are attached to a surface and they can't move. Sponges do not produce their own food, so remember that terminology there was autotrophs, so these guys are heterotrophs, right? Instead, they are filter feeders, and when we refer to filter feeders, we've said that they are cleaning. So how do they clean? Well, they obtain their food by straining the water through their pores, okay, for small bits of food like bacteria, algae, protozoans. Sponges exhibit less specialization of cells than most invertebrates. They have a mesophyll, which is just, they have two layers, the endoderm and the ectoderm. Again, when we're talking about development in the germ layers, these guys only have two, and then there's a layer in between the endoderm and the ectoderm, and that's a mesophyll layer, and it's basically a jelly-like substance. The outer layer of the sponge is the epidermis, and these are flat cells which are made out of epithelial tissue, or epithelial cells, sorry. The inner layer has a gastrodermis layer, and they basically consist of collar cells. So what is that? Collar cells have flagella in them, and the flagella is what will move water through an incurrent pore. So remember when we were talking about bivalves, we talked about incurrent and excurrent siphons or pores. Here we call them an incurrent pore, and water then travels out the osculum at the top of the sponge. So if you can picture this just being a sponge here, okay, or in between my hands there on either side, all right, I have pores all throughout. You know what a sponge looks like, so we have pores all throughout the sponge, and that water is flowing through the sponge, and basically as nutrients are grabbed and whatever, any waste that is going to leave that pore is going to go out through the osculum, and it's going to be shot out like rather quickly. All right. Let me see. Mesophyll between the epidermis and the collar cells are called mesophyll between the epidermis and the collar cells are called ambiocytes because they look like amoebas. And the job of the amoebocyte is basically to travel around the cell, distributing food and oxygen. And so if you ever seen the a slide of an amoeba, actually it just crawling around, and it is just looking for something to ingest, basically. And so again, the amoebocyte, C-Y-T-E-S at the end of that word there, amoebocytes. So it's a cell, an amoeba cell, that is looking to ingest food and oxygen. All right. Because the let me see. Yeah, no, that's fine. So let's see, the body of the sponge. Okay, so if the sponge didn't have specific structures, it would basically collapse. So what are the specific structures that allow the sponge to remain erect in the water? Would be that the sponge has spongin and spicules. So some sponges that have a soft network of protein fibers are spongin. Okay, they're softer, but the spicules are harder, and many of the spicules stick out of the epidermis, and that provides protection for the sponge because they're harder, and so it wouldn't feel as good for an organism that might be trying to prey on a sponge. It wouldn't feel good if they brushed up against the spicules. So it's a defense mechanism. It has a defense mechanism. The combination of the spongin and the spicules will determine whether or not the organism is softer or harder. So if it has more spongin, it's going to be more soft, and if it has more spicules, it will be more hard. Make sense? All right. Reproduction of the sponge occurs both asexually and sexually. So there are 3 ways for a sponge to actually reproduce asexually, and it's through budding and through regeneration. And let me just see, sponges can simply reproduce by budding, where a new sponge grows from the older one, and then we saw that again with the polyp, and again that will just break off. And sponges reproduce by regeneration, where just a small body part is broken off and regrows. Okay. And then people harvest sponges, often taking advantage of the breaking off pieces of a catch and throwing them back into the water to harvest for later. And then finally, sponges can reproduce by gemmules, which is a group of ambiocytes that are covered by a hard outer covering. Gemmules are released from the osculum after the adult sponge dies. All right. So 3 ways: it's through budding, through regeneration, and through gemmules. And then lastly, they can reproduce. Those are 3 ways asexually that they can reproduce. But then lastly, sexually, they can reproduce when a sponge releases sperm into the water, and then the sperm travels to another sponge and fertilizes its egg. The larvae from them then swim to another location using flagella. Okay, so that whip-like motion, or a tail-like ending. It has a tail-like end. We use that flagella where it will grow into a sponge. And then most sponge species are hermaphrodites, meaning that they contain both male and female parts to be able to reproduce. That's the sponges. I will come back later on with a video on the last part of this section, which is the annelids, the segmented worms, and I think we have 2 sections, I'm sorry, and then the protozoa. And then we're going to wrap up chapter 21. But I actually have to get going, so I will get that next video out shortly.